The last inheritance pattern is called multiple alleles. So in multiple alleles, you can have not just 2 letters, you end up having 3 or more. Now, pretty much the only example we're going to be using here is blood type. You may have heard of A type blood or O type blood. We also have AB type blood, and we also have O type blood. So let's explore a little bit about what that means. So for blood type A, I'm going to use a red color here for blood. The phenotype is A. Possible genotypes. A is actually a dominant allele. So in the case of blood type A, it could have 2 A's, or it could have an A and an O, because type O is recessive. Now, I like doing it like this, where I have capital A's and capital B's, and then lowercase o's, because I know that these 2 are dominant and this is recessive. Another way to do it, which you don't have to, is to use an I, and then the A goes on the I, and a lowercase I for the O allele. Or you could have 2 A's, where I has an A exponent. Now, I here represents having the antigen, and the dominant version of that. I means, lowercase I, recessive, means no antigen. So if I have type A alleles, I'm coding for something on the outside of the blood, and it's like a little protein, signaler, receptor, something on the outside of the cell that lets your body know who they are. So that's why it's important you get the right blood type, because if your body doesn't recognize those kind of signaling cues, then your body would react to that as some sort of outside invader. So if I have type B blood, type B is my phenotype. Genotype, I could have 2 B's, or I could have just 1 B with the recessive O. If I have the I's, again, type 2B alleles, or just the 1 with the type O, lowercase I is recessive, that's the type O. Type B gives you a different receptor signaler on the outside. So the body would recognize or not recognize this different receptor. So type A people can't really give their blood to type B people, because type B people wouldn't recognize this on the outside. And that their body would react, and it would coagulate, cause some blood clots, and it could be very bad for them. As you can imagine, type AB phenotype is type AB. Possible genotypes? Well, we only have 1 for this one, having an A and a B allele. Now, the I version of that, 2 capitals with the A and the B exponents. Type O is recessive. So we have 2 O alleles. I like to keep them lowercase. Excuse me, that's genotype. O is our phenotype. Genotypes, 2 O alleles, or 2 lowercase i's as the other version. I'll put all the I ones in a box so you know that they mean the same thing. I like to just stick with the simple ones though. Let's add our both A and B antigens go on the outside of AB blood. So it'd be hard for these guys to not recognize something. They could recognize A blood that got donated to them, or B blood that got donated to them. Now O doesn't have anything on the outside. It doesn't code for an antigen, so there's nothing there. O people can give their blood to any of these people, but if any of these people give their blood to O, there's all these things on the outside that they wouldn't recognize, and their body would react. So what does a cross look like? So my trait here, again, is blood type. I've got A alleles, I've got B alleles, and I've got O alleles. My genotypes could be A, heterozygous A, B, homozygous, heterozygous B, type O, or type AB. All those are possible genotypes. And the phenotypes are type A, type B, type O, or type AB. Let's do a test cross for some heterozygous people again. Let's do heterozygous for type A, heterozygous for type B. This will give us a lot of options. So I've got one parent up here, the other parent over here. A and B go in this box. B and O go in this box. A and O go in this box. O go in that box. So I have all my options here. All blood types would end up being a result of this cross. So I don't have any homozygous type A. I've got one heterozygous type A, no homozygous type B, one homozygous type B, one type O, and one type AB. So I've got 1 to 1 to 1 ratios here. I've got 4 different possible phenotypes. So you add more alleles, you get more phenotypes, and you get more possible options from your results. On the back side of this paper, you'll see some practice problems here. First questions just ask you to figure out some genotypes and phenotypes, and then number 2, 3, and 4 will ask you to set up some crosses, so you'll make your Punnett squares.